Our seventh speaker is Sushobhna Batra from Dr. Mark Diamond's lab, and she is in the immunology program. If this 3-minute thesis gig doesn't work for her, Sushobhna will give stand-up comedy a shot. And this is not a joke. Her talk is entitled Alzheimer's Disease, A Tale of a Tau Tangle.
So think back again when you last forget where you left your car keys or couldn't remember your own cell phone number. Now, imagine the same scenario, but it gradually becoming a permanent part of your life, constantly forgetting the little things that are important details of your life. This forgetfulness is the primary symptom that patients with Alzheimer's disease live with every single day. But what is really going on within the neurons of these patients' brains that results in this loss of memory function? The answer lies in the aggregation of a protein called tau. In healthy brains, tau is just existing in a soluble form, hanging around with the cytoskeletal structures of components of the cell. In disease, however, tau forms these spaghetti-like clumps within the neurons, which leads to their death, and that manifests as cognitive decline over time. These tau aggregates can in turn serve as seeds of propagation, templating onto the monomeric physiological tau in a prion-like manner and cause more tau to continue to aggregate. Remember the saying, one rotten apple can spoil the bunch? So does an aggregated tau, by recruiting healthy fragments of soluble tau into growing clumps of tau tangles over time. It seems natural that the cell would have some mechanism of detecting these tau aggregates as cellular garbage and getting rid of them as soon as possible. Understanding the cell's role in this initial stage and processing of tau aggregation is the very quest of my project. To do this, I basically labeled and detected cellular factors in the vicinity of tau right as it started to aggregate, and this led me to identify a single highly enriched protein called the valosin-containing protein, VCP. So what's VCP doing in this whole story? Let's go back to the the rotten apple analogy once again. A simple way to get rid of this rotten apple waste is we pass it through the kitchen sink's garbage disposal system, which will break it down into tiny bits and pieces. VCP is your cell's garbage disposal equivalent. My work using cellular models of tau aggregation have shown that VCP can chop down the bulky aggregates of the tau protein into tiny fragments. It does so by almost acting like a machine that will thread through or unfold these little pieces of tau fibrils from a giant clump and then sends these fragments for removal through the cell's degradation machinery. I'm now looking into investigating the precise mechanistic details of how VCP and its related pathways can modulate tau aggregation, and thereby identify new therapeutic targets to intervene this aggregation cascade. This is essential to study because by curbing the tau aggregation process, we can help eliminate the underlying root cause of Alzheimer's and thereby contribute to a permanent cure of the disease, a treatment that currently does not exist. Thank you.